Welcome to the Medical Science Concepts by Dr. U.U.T. Umair Usmani Tolkhi. Okay, so in the last videos we discussed both in English and Urdu the introduction, then we discussed the alkylating agents, then we discussed the chemotherapy resistance, and in this video we will discuss in English the anti-tumor antibiotics. So for Urdu version you can search us anywhere you want, Facebook or YouTube. So anti-tumor antibiotics. We have antibiotics that are used for the treatment of tumor, or you can say anti-tumor antibiotics. So we classify them into two categories, one is anthracyclins, other one is bleomycin. Anthracycline is further divided into 1, 2, 3, 4, 5 or how many, but I remember two, doxorubicin and daunorubicin. These two are the antibiotics that are put in the category of anthracyclins, and another one is bleomycin, which is also used as an anti-tumor antibiotic. Okay, before we go to these, let us understand the general mechanism of action or the general mode of action of antibiotics. What they do is, first of all, they inhibit the topoisomerases. You know, topoisomerase 1, topoisomerase 2. These two are involved in a replication process when DNA is going to divide into two. In that, while topoisomerase 1 and topoisomerase 2, they are supposed to act. What are they doing? They are unwinding and winding. Topoisomerase 1 is supposed to unwind or wind only one strand, and whereas topoisomerase 2 is supposed to unwind or wind the two strands of the DNA. That's it. So what antibiotics do is that they inhibit the topoisomerases. When you inhibit the topoisomerases enzymes, then what is going to happen? No replication. So if our cell is not going to, our DNA is not going to replicate, then no cells will be formed. Okay, so like that we inhibit the process of replication of this DNA of the tumor cell. Like that we can counter the cell division of the cancerous cell. And it produces the free radical. You know, free radicals, just I'm giving you an example of hydroxyl ion. When a dot is written at the top of the hydroxyl, then this is given the name as a radical. According to chemistry point of view, when there is a dot, it means that this is a free radical, which is very active. It is no charge, neither positive nor negative. It has a valence electron pair that is going to attack any atom or any molecule or any ion or any compound in a very drastic way, in a very strong way. So this hydroxyl ion, or this is not an ion, this is a radical. This radical or any other radical that is formed will go and will take the phosphodiester linkage of the DNA, where specifically if we talk, it will take the phosphate. You can say the phosphodiester linkage we often understand with the phosphate. So at the phosphate, this hydroxyl, this radical will attack. After attacking, it will destroy the structure of the DNA. So like that, we counter the structure of DNA. When the structure of DNA is no more available, then there is no more cell division. Like that, the antibiotics have their mode of action. Now, we discuss the anthracycline. How anthracyclins they do? Doxorubicin or daunorubicin. What are they doing? They first of all they undergo the lipid membrane peroxidation. When there is the peroxidation process done on the lipid membrane, you know, a cell is made up of a lipid bilayer. So when this membrane is undergoing the process of peroxidation, so your membrane is going to be destroyed, and this membrane gets destroyed. After that, these daunorubicin or doxorubicin, or you can say anthracyclins, are also involved in the peroxidation of the purines, pyrimidines, thiols, amines. You know what purines and pyrimidines, which are the nitrogenous bases. When the structure of purines, pyrimidines, thiols or amines, when their structures are changed by means of oxidation, the addition of oxygen or by the loss of electrons from them, then what will happen? They will be no more able to undergo the process of replication or the formation of DNA. So our main target in anti-tumor is to stop the cell division by any way. If you are going to stop, if you are going to change the structure of purines and pyrimides, nitrogenous bases, which will be no more able to combine, like adenine, thymine before they were able to combine. When you do the structure modification, when you change the structure of adenine, guanine, cytosine, thymine, then they will not understand where to bind. Like that, you stop their mechanism, their binding mechanism, you stop that. So when you destroy the cell membrane, again we got the very interesting thing that we are supposed means our cell is no more able to live. When the cell is not anymore able to live, when that get die, so then what will happen? We will be able to counter the cancer. And we'll discuss the bleomycin in our next video.